Astrocytes have an important role in controlling oxidative stress of the central nervous system, helping maintain a homeostatic environment for neurons, as well as protecting neurons from reactive oxygen species. Here, a method for assaying oxidative stress in astrocytes is presented. To illustrate how this assay works, hydrogen peroxide, an ROS typically produced during cellular metabolism, is applied to feline astrocytes. Next, to assay for reactive oxygen species, the cell permeable indicator CMH2-DCFDA is added to the cells. CMH2-DCFDA enters the cell as a non-fluorescent compound. There, cellular esterases remove the acetate groups. If reactive oxygen species are present, the compound is oxidized and becomes fluorescent. The cells are then harvested, and fluorescence is assayed by flow cytometry. The number of fluorescent cells indicates the number of cells that are in an oxidative state.
The aim, of course, is then to detect which specific oxygen radicals are present in the cells by using different assays. This is particularly very important in neurology and basically paved the way for new avenues of research in neurology, because we can basically relate different diseases or different agents with different radicals produced in a neurological disease. Part of this job is due to the technique mastered by Omar Alvarado, Andrea Wournell, which are our two lab technicians, wonderful, and our great Jonathan Lee, which is a graduate student.
Begin this procedure by culturing the feline astrocyte cell line G355-5 in a 70